recently. Suppose this is a bacteria, and on top of the bacteria is a virus, that is a bacteriophage, a virus that infects the bacteria. So the virus is on top of the bacteria, and this green that you're seeing is the genetic material of the virus. And this virus will attach to the surface of bacteria, and it will inject its genetic material into the cell. Once inside the cell, this viral genetic material will integrate with the bacterial genome. The red is what is bacterial genome, this one. So it will integrate with the bacterial genome, and it will begin to produce viral proteins. That is how the virus infects the bacteria. Now, it was seen that some of the bacteria, they can resist the infection by such bacteriophages or viruses. So basically the whole CRISPR came up with a question, a basic question: how is a bacteria able to fight, or how some of the bacteria are able to fight these viruses? And in 1987, Japanese researchers found that the bacterial genome had repeating areas of similar repeating repeats with dissimilar genetic material in between. This is dissimilar, and repeats of genetic material were seen. That was found in 1987, and that was that, and they left it at there. They didn't understand what it was. Later, and it was called CRISPR, that is clustered regularly interspaced palindromic repeats, which means if this is a genome, this was seen in clusters. In here it may be seen here. So such material, such repeats were seen in clusters in the genome, clustered, and within the clusters, and that is magnified here, within the clusters there were regularly interspaced, see, it is regularly interspersed repeats, and these are the repeats. The white is the repeat, and they were palindromic. That means the base pairs, if you read from here, this direction or this direction, it is the same. That is what you call a palindrome. So it is clustered within the genome, and within the clusters there were regularly interspaced palindromic repeats, and in between the repeats were spaces, and these spaces, they varied from 1 to 1, and they were never the same. Between the repeats were seen the spaces. Now we'll come to the Cas genes later and all that, but now, as of now, that is what a CRISPR was or is. Now in 2007 it was found, or it was thought to be a form of adaptive immunity against viruses, and that was basically discovered or proposed by a dairy company called Danisco, and it was put forward that it is the spaces that you see, because they are different, each is different, the colors are different. So the spaces represent the infections that this bacteria might have encountered previously. That is, the bacterial genome will have this repeats, and within the repeats, in between the repeats, are spaces that are thought to be viral, or that were proposed to be viral genomes. So that means it is a chronological order of the infections that this bacteria might have experienced in its lifetime. Now coming to spaces. Spaces are nucleic, as already told, nucleic acids of viruses that infect the bacteria. Now if new spaces are added, that is, new viruses are recognized, which means if a bacteria has a particular spacer, suppose this spacer is present in a bacteria, and a virus is going to attack this bacteria, and that virus has the same genetic material as seen here, then this bacteria is able to recognize that virus. So if a bacteria can get more and more spaces, that means it can recognize more and more different types of viruses. It also means it has encountered all those infections in its lifetime. That is, the newly added chronologically to one side of CRISPR. So we can see it is a chronological order, so we can know which infection was first and which was last, as seen in this image. Suppose this is a bacterial genome with the CRISPR locus of the bacterial genome. We can see the repeats. These are the repeats, the similar areas, and these show the previous spacers. And this is a new spacer, that is a new infection by a virus, and that viral genetic material is added onto this area. So that's a new spacer. And with time, this is time, as time is going, it is getting another infection, and this is another infection by another virus. So this gets added here. So this was the first and this was the second. Not first, here we have previous, but as far as this area is concerned, it is first and second, and this is the third. So we get a chronological order of the infection this bacteria might have encountered in its life. So now coming to CRISPR, it was seen that there are 3 types of CRISPRs that is operating in the bacteria and archaea, and we are concerned only with type 2 because that is what is most commonly used in genome editing or gene editing tools, etc. And we'll only talk about that hence. And coming to the type 2. Type 2 CRISPR, this is a CRISPR locus. It has Cas genes, tracer genes, and an array called Crispr array. Now we'll be discussing about all that. So we have already told within the clusters we have the CRISPR loci. This is the CRISPR loci, and within the CRISPR locus we have this repeats. The white is the repeats, and these are the unique viral genomes that the bacteria attained from each infections. And adjacent to the CRISPR locus, this is the CRISPR locus, we have Cas genes or CRISPR associated genes, and near the Cas genes we have another set of genes called tracer genes, and the tracer genes will encode or code for the tracer RNA, and the Cas genes will code for a protein called Cas9, and the CRISPR array, they will code for pre-CRISPR RNA or pre-CR RNA. And this pre-CR RNA has the repeats and also several of the unique sequences of the viral genome. So RNase 3, the enzyme RNase 3 will, once this pre-CRISPR RNA is attached to the Cas9 whole system along with the tracer RNA, the RNase 3 will cleave this pre-CRISPR RNA to CRISPR RNA. That is called maturation. So for maturation of pre-CRISPR RNA, we require RNase 3. So RNase 3 is important for maturation of pre-CRISPR RNA to CRISPR RNA, and this CRISPR RNA contains this tracer RNA. This blue is the tracer RNA, then this white is the repeat area. One repeat, one tracer RNA, and one unique sequence of the virus. And it has to be remembered that the tracer RNA will attach only to the repeats, that is the CRISPR repeats, not to the unique area. This unique area is for attached, that will come to later. Now coming to Cas9, it is a protein, and it is a protein and it acts as a helicase as well as an endonuclease. Now it requires a PAM sequence in the target, that is in our case a virus. We'll come to the PAM sequence later. Anyway, coming to the Cas9, it is a protein that is central to the type 2 CRISPR system, and it can act as a helicase in, and that is it will unwind the DNA, the viral DNA, and it is also acts as a endonuclease, that is it is able to cut the viral DNA. So now tracer RNA or transactive activating CRISPR RNA. It is a small helper RNA and it plays a role in maturation of CRISPR RNA and helps in CRISPR RNA Cas9 activation. And this is the tracer RNA here, and it is required for the activation, and it can be seen before that is upstream of the CRISPR locus. This is the CRISPR locus, and this is the tracer RNA gene. And this has, as already told, it has a repeat, it has a region complementary to the repeat region. The white is the repeat, and the blue here we can see the white is the repeat and blue is the tracer RNA, complementary to the repeat, and it will form pairs with that. And the tracer RNA also associates with the Cas9 protein, that is the Cas9 protein. Now next part of the equation is pre-CRISPR RNA. We have already told about the pre-CRISPR RNA, which is a long precursor of the CRISPR RNA, and RNase 3 is required for the maturation of that to CRISPR RNA. Now, these are sequences that is present in the virus or the viral genome. This CRISPR is a locus that is seen in the bacterial genome. In the bacterial genome, CRISPR is a locus that is seen in the bacterial genome. Now we are coming to the genetic areas that are present in the virus or target DNA that is required for the system to act. So first one is protospacer, and second is PAM sequence. We already mentioned about PAM sequence once, but we did not say what it was. Now protospacer is the part of the viral DNA that is complementary to the CRISPR RNA. So if this is the viral genome and this area, it is complementary. If it is complementary to the CRISPR RNA, CRISPR RNA means the unique area, the spacer area. This is the spacer area of CRISPR RNA. This maybe just imagine this to be the repeat area, and this will be the tracer RNA. The color is not that, but just imagine that. Anyway, so this is the protospacer. So the protospacer is the part of the viral genome that base pairs with, or that is complementary to, the spacer of the CRISPR RNA, or the what do you call, the spacer part of the viral genome, bacterial genome. Now coming to PAM sequence, it's also called that is full form is protospacer adjacent motif, and that is a sequence that is adjacent to this. Let's change the color of the pen. Okay. So this is the PAM sequence. It is a 3 base pair sequence, and it is adjacent to the protospacer. This is the protospacer which binds to the CRISPR RNA, and adjacent to that is the PAM sequence, which is a 3 base pair sequence in the sorry, it is not here, it is only seen in the viral or target area, not here. So this is the PAM sequence. So PAM sequence is required for attaching Cas9 protein to the viral genome. Now coming to the defense, how the bacteria is able to defend itself from the virus. There are basically 3 steps and can be called, first is the adaptation, second is expression, and third is interference. Adaptation is referring to the insertion of new spacers. That is, when a virus infects a bacteria, it will provide the genetic material to the bacteria. That is what you call a spacer. So the bacteria gets a new spacer. That is called adaptation, and it will provide genetic memory. That is, it acts like an adaptive immunity. We'll come to that soon. Then expression, that is a second step in the defense, and that means the production of CRISPR RNA and the maturation of that to CRISPR RNA. Then comes interference. Interference is what is most important. That is basically the cutting up of the viral genome in case of bacterial infection. And once the genome of the virus is cut by the endonuclease, the virus genome will disintegrate. Right. Now this image shows the whole thing, and it is quite congested, so I'll just explain here. This is the bacterial cell membrane, and this is the virus that is attaching to the cell membrane. This is the viral genome, and we'll concentrate in this area first of all. And here, this viral genome is what we see here. Double-stranded viral DNA is injected into the bacteria. And don't look at this. Don't look at this. Look at this. So the double-stranded DNA of the virus is injected in the bacteria, and here that is injected, and we can just let it be there. Now we'll go to the Cas. Now the Cas protein is here, and this Cas protein present in the bacteria will go and bind to the viral double-strand DNA, and a part of that is taken, and a new spacer, a novel spacer is formed from that. And this is the new spacer that we got from this double-strand viral DNA, and a new spacer is formed, and this spacer is incorporated into the CRISPR array. This is a CRISPR loci and the CRISPR array, and these are the repeats with spaces in between. And here we are only showing the new one, that new spacer. So anyway, the spacer gets attached to the CRISPR array, and then this spacer gets transcribed and processed to CRISPR RNA. And this spacer along with the other tracer RNA, etc., will go on and form a complex with the Cas CRISPR RNA complex. Now that is here. Now imagine the same virus is infecting the person again, sorry, the bacteria again. Now this is a different scenario. The first scenario was the virus infected, its DNA entered the bacteria, and a novel spacer was formed, and the novel spacer was transcribed, and it is sitting here and ready to act here along with the Cas. Now this is a second virus, the same virus or the similar species of virus, or whatever you call that. It has infected it again, and in this is the first and this is the second time it is coming. And when it comes, this sequence is recognized because it is complementary. This this spacer is complementary to that virus, so this will recognize the virus as the previous one, and once the complementary strand is hybridized or base paired with the viral double-strand viral DNA, it is cut by the endonuclease in Cas, and the viral DNA is inactivated. So that is how a bacteria will destroy or inactivate a virus with the CRISPR system. Now this is a system which some scientists thought, how can we manipulate that and use that for other genetic engineering purposes. So basic thing is we have to have, or basic thing is the CRISPR locus. In this CRISPR locus we have to have a sequence that is here, that is complementary to a sequence here, and if the sequence in the CRISPR is complementary, that is here, if this sequence is complementary to another DNA that is coming into contact, then that DNA will be cut by the Cas enzyme. So that is the basis of genetic engineering, genetic editing, genome editing. Now genome editing, we can divide it before CRISPR and CRISPR. Maybe something will come after CRISPR, that we don't know. As of now, before CRISPR we had zinc finger nucleases, talons, etc. And the problem was they were all very costly, and not every lab could afford that. Also, they needed a new protein for each experiment. For each DNA sequence that had to be targeted, they had to produce, or they had to prepare a new protein, and producing a new protein is very labor intensive and very difficult. So that was not or it did not gain much of a wide recognition, or much of people could not accept or use that in their labs. And the advantage of CRISPR was it is comparatively low cost. It's not that it's dirt cheap, it is comparatively low cost, and we don't require to produce a new protein for each DNA sequence. We only have to design an RNA, which is a quite pretty easy. You can do in a single day in labs, and in some cases it only takes hours to prepare or design an RNA which is complementary to the DNA strand that we require or that we want to edit. So it is comparatively low cost and low time consuming, less labor intensive method, and it gained wide acceptance. Now we have already told about the advantages of CRISPR. That is the Cas9 protein, we don't have to do anything with it, it is fixed. Then the RNA component is changed. That is what we told. It is a 20 nucleotide sequence that we have to change. Only the 20 nucleotide sequence corresponding to the target, complementary to the target DNA site, we have to change for each target, and the Cas protein is same for all. Then it has a multiplex capacity, which means we can inject multiple CRISPRs with multiple targets, with different targets. We can inject all of them together into a cell, and each of them will go and act, and we can edit in different areas at the same time. So it is a multiplex it has a multiplex capacity compared to the previous generations, which we could only edit one at a time. It is very difficult. So this is the Cas9 protein, and this is our tracer RNA, and we can see again the tracer RNA base pairs with the repeat area, CRISPR repeats. The white is the CRISPR repeat, and this is the complementary strand that pairs with the target. This is our target. Now we are telling target and not virus, because this is our target. We are not talking about bacteria now. Now this is the target DNA, and if you look the target DNA, this is the PAM sequence. The PAM is a 3 nucleotide. You can see 3 base pairs. 3 nucleotide sequence seen adjacent to the protospacer. This is the protospacer. Protospacer is the part of the target DNA or the viral DNA that base pairs with the spacer. So this is the spacer. This is the protospacer. Spacer, protospacer, PAM sequence, which is the PAM sequence is the part to which the Cas9 protein attaches. So I think it's covered, right? Now there are certain rules in all of this, and the protospacer, again I'm telling, this is the protospacer. Protospacer should be 20 base pair long, which means the spacer is also 20 base pair long, which again means we only need to change this 20 base pairs if we want to target a different site. Alright, now the protospacer is just before PAM, and PAM, in case of Streptococcus pyogenes, which is the most commonly used bacterium in CRISPR experiments, and in that it is always NGG, the sequence, and N stands for any nucleotide and G guanine, and it is already told is a protospacer. So the protospacer in Streptococcus pyogenes is NGG, and only if that is present, this Cas9 can go and attach to the genomic DNA, right? Then the PAM is recognized, the PAM sequence is recognized by the Cas9, and without PAM we cannot form this complex. Let's change the color of the pen to something maybe not red, okay, blue. So without the PAM sequence the complex cannot form, because the Cas9 protein attaches to PAM. Now once this attachment occurs, there is a break in the double strand that occurs 3 base pairs upstream of PAM. This is the PAM, and 3 base pairs upstream of PAM we get a double strand cleave or break. So again, this is a tracer, this is a this is the CRISPR locus, this is the tracer, this is a Cas genes, tracer RNA, Cas protein, Precursor RNA, process to CRISPR RNA. Just a revision. Now here we can see the Cas9 protein, to which we have attached the tracer RNA with the spacer. This is a spacer RNA along with the repeat, which is called the CRISPR RNA together. And now it has gone and attached to a genomic DNA or a target DNA, and this is the PAM sequence that is seen in the target DNA, the genomic DNA, or in case of bacteria, the viral DNA. In case of bacterial infections, infection in bacteria, viral genome. So the genomic DNA or the target genome DNA has the PAM and the Proto spacer, and this is the CRISPR RNA tracer. Okay, and now once that occurs, there is a cleavage 3 base pairs upstream of PAM sequence, and the DNA will cleave. Here we can see the cleaving of the DNA, and this genomic DNA has cleaved and a break has occurred here. You can see the break has occurred, and once the break occurs, it has to heal back or anneal back, because DNA cannot survive with a break. It is not possible for a survival. So two methods are there. One is non-homogeneous homologous. One is non-homologous end joining, and the other is homologous directed repair. Now here we can again see this is the genomic DNA, and the break has occurred. It can either repair by non-homologous end joining, and non-homologous end joining is a very error-prone mechanism. That is, it is not foolproof, and it can have when this two ends join back together, it can have certain insertions or deletions. Both insertions as well as deletions can cause frame shift mutations or other gene disruptions, etc. So this is if there is a gene that produces any disease, and this insertion originally if there was a gene that produced a disease here, and we are able to insert or delete a part of that gene, that means we can silence that gene. So that gene is disrupted. That amounts to genetic editing or gene editing. Now coming to homologous directed repair, this is more of a engineered version in which we can add on whatever features that we need. If we need a certain feature, we can add that into the experiment. That is, once the break has happened and we want to add this template, this double strand DNA template to this area, we just have to add this template into the culture medium or the wherever the experiment is done, and that template gets added on to the area where the cut has occurred, where the double strand break has occurred. So that means a new sequence is formed. A new sequence means there is new genetic features. So it is like we can add any feature to a person, and suppose if this area represented blue eyes, and this is added on here, this person will have blue eyes, etc. I'm just telling a wild example, I'm not sure. Anyway, this is what you call knock-in. Sorry, knockout. Knockout is when you knock out an expression that is already present due to a gene, and expression of a gene is blocked or removed. That is knockout. Knock-in is when you put in a desired genetic expression into, or a desired gene sequence into the double strand break region. That is called knock-in, so that there is a new expression. So knock-in and knockout. You might have heard of the knockout mice. Now coming to non-homologous end joining, as already told, it is a random process. There is random deletions, random insertions, that is called indels, very error prone, and it can result in frame shift mutations, and that is knockout. And in case of homology directed repair, it is a precise modification. We can add a precise genetic material that we require to be added. Then we add as a repair template. We give it as a repair template whatever feature that we need. That is called knock-in mutation. Now coming to the applications of this CRISPR, it is vast, and in fact we are not yet sure how much application is possible. It is increasing day by day. Anyway, it has tremendous application in human medicine. In case of human medicine, it is being tried for sickle cell disease, for thalassemia, then what do you call hypertrophic cardiomyopathy, then leukemia, cancers, HIV, and the list goes on and on. So it's being tried for almost all the disease conditions that is possible in humans, and it is used in agriculture to produce disease resistant crops, to give new features, etc. In fact, it was first, as I already told, in 2007 a dairy company found out about the immune function of the CRISPR locus, right? Then it is used in biotechnology, it is used in biological research, etc.